And so the title of the talk that I'm going to give is From Complexity to Clarity, Building 40 plus parameter panels for imaging with ease. If you are attending this talk, you're probably pretty aware that there is a high degree of heterogeneity in pretty much every biological system. And to identify the need for this, I like to borrow from Jared Burks at MD Anderson. Jared published this Cell Gene Therapy Insights publication in 2021. And he had this figure that I've always loved. It sort of relates tissue to a neighborhood. And we need to have some of those same considerations. So if I'm trying to drive my car or my cells through a neighborhood, I need to understand the pathways, and I need to understand that maybe there are different regions within that city. Where are roadblocks? What are the other immune cells and how they're responding? Where are we getting immune suppression, et cetera? And so spatial information becomes critical in defining disease and especially disease treatment. Now, this is not a new concept. The concept of being able to look at space and tissues has been around for quite a while. If we think about hematoloxin and inosine, this was developed in the late 1800s and had developments along the way, like chromogenic staining in the early 1900s, that can help move us forward. But this is still considered to be the gold standard for a lot of clinicians when they're assessing tumor or disease state and grading that particular progression of that particular tumor throughout that tissue. So the idea is that you would use hematoloxin, right? So you get the ability to determine where your nuclei are with this purple stain, or you may use an acidic dye like inosine to sort of find the basic structures. And so you're looking at these shades of pink to red, and a pathologist has to come in and score that tissue. And while I'm not saying that we won't need pathologists, we will always need pathologists. More information is always better. And let's say I wanted to look at one particular target and I wanted to employ an antibody, I can then do a chromogenic staining and I can look at perhaps CD8. The problem with that is I'm limited. What if I want to look at CD3? Then I have to do a serial section, and perhaps now I'm doing another serial section to look at CD20 and so forth. Now microscopy has aided us in the fact that we can do 3 to 6, maybe now 7 or 8, and with the really advanced systems that almost no one has access to, upwards of 20, but you're still limited. But the idea is maybe we can start looking in a single field of vision for more antibodies simultaneously. And this is where our technology really shines. Now, I'm going to spend a little time talking about what distinguishes us from everyone else. We're antibody-based technology, and we use metals that are tagged to antibodies instead of fluorophores. Now, fluorophores have been around for a long time. I'm not going to discount fluorophores. I, in fact, did a lot of use on microscopes and use of fluorophores in flow cytometry. But the potential problem you have in the high-plex space is the fact that fluorophores are not just in one wavelength. Let's say we were talking about Alexa fluor 488. We know that Alexa fluor 488 is not just in the 488 nanometer space. There's a broad spread in that particular spectrum. And every time you add a fluorophore, you have to worry about the spread of that individual fluorophore into adjacent channels. And so in order to deal with that, there are things like spectral unmixing or compensation. So you need a lot of controls. And you also have to consider in those cases, if that control then needs to be repeated over time due to the change of that fluorophore in space. Our technology allows us to tag a specific metal to an antibody and have 1 Dalton resolution. That means we can detect on our technology a mass range anywhere from 75 Daltons to 210 Daltons, 135 mass channel. So if I have neodymium 150 and I want to tag it to another samarium 151, I can easily put those, even though those metals are right next to each other with 1 mass. And so we have a lot of room to grow. So we get very clean data when we're moving through our data with very little background. This figure is not one that we produced. This is from a spotlight from Bowden Miller's group back in 2022. It simply illustrated, in this case, he was using pan-cytokeratin, using IMC, Imaging Mass Cytometry, our technology. What we can see here is very tight, succinct expression of pan-cytokeratin in the very clean viewing of the macrophages underneath expressed by CD68. And however, if you compare that to immunofluorescence, that autofluorescence can mask a lot of that CD68. And so the advantage is having very clean signals, which allow us to have a very big dynamic range. Our dynamic range can approach 1 × 10⁵, where fluorophores are usually somewhere in 1 times 10 to the 2 or 1 times 10 to the 3 at best. In addition to that, we are non-cyclic technology. We can stain all of our antibodies at one time, and then we can get that information. And so how others have developed a strategy to get around that autofluorescence is to limit the number of targets to maybe staining the entire panel of antibodies, but then using secondaries in 2 or 3 at a time and doing cycle or a cyclic approach to build upon those fluorophores that you're using over and over and over, and then overlaying those images together. That strategy employs potentially more time, more cost, and certainly with extra rounds, you will see some level of damage. It can be high, it can be low, depending on the number of cycles you're employing, but we do not have that issue. And so if you're going to encounter that damage, you have to ask yourself, have I noted which markers are going to incur, which markers are going to be lost first, and which ones are going to be lost later, so I can sort of design that in my experiment. These are not considerations that we have to worry about. We can pretty much design our experiments without having to worry about the order of anything. So the idea is we have a system that gives you end-to-end workflow from designing panels, from the tools and resources we have, single staining, analysis tools. We can pretty quickly move you very quickly to results from the very beginning. We have a lot of reagents for both human and mouse. We have over 300 antibody catalog antibodies. We have conjugation kits that allow you to conjugate your own antibodies. We have in-house custom service that will do conjugations for you. We have MaxPar on demand. So if you see that we have a specific antibody, but it's not on the metal that you want, we can change that metal for you with the service. We have a number of different things for both mouse and human, but the majority of the last part of this talk is going to be focused on something that we're really excited about, which is our kits. So we've developed a number of different kits that sort of allow you to move very fast into the high parameter space. Now, if you're building a panel, we want to make sure that we include markers for every area of biology, from structural to phenotypic to functional. Now, when we design a panel, we keep all of this in mind. And so the first panel that I'm going to introduce to you is our human immuno-oncology panel, which is 31 markers. So if you're in the immuno-oncology space, we have a panel that is already by itself in high-plex. But we have those considerations of structural markers. So let's say we're looking at structural. We have 3 different modules: epithelial mesenchymal, tissue architecture, stromal cells, with a number of different markers. I'm not going to go ahead and read all of the different details, but you can clearly see the markers we have in each of these panels. We also want to make sure we cover phenotyping. So we have basic phenotyping antibodies, lymphoid antibodies, and myeloid antibodies that are included, all in a modular set. And of course, we have a module that covers the functional state. So if we're looking at regulatory T cells, we have FOXP3. And of course, if we're looking at maybe NK cells, granzyme B, or other cells that kill, or proliferation, or some immune suppression, we have a kit that we think will cover the biology for most people who are interested in immunology, as well as oncology. But that is not the only panel that we have. Even though that alone takes you in the high-plex space, the title of this talk is how do we move into 40 parameters or more. And so the idea is that we can use this first panel potentially as a building block or foundational panel, and then use other panels or other antibodies to sort of expand into that space. T-cell exhaustion, glioblastoma, neurophenotyping or expansion, as well as a segmentation kit, and the additional antibodies from our catalog can all be used foundationally to grow your panel. So let's take a look at that in an area of immuno-oncology. So let's say we're going to build this panel with 31 and we want to expand to 41 parameters. We can simply employ, in this case, our human immune cell expansion panel, as well as our segmentation kit, and then we're at 41. Now, in this particular case, let's use an example. Here, we took this same exact panel and we stained breast cancer. Now, we used preview mode. Now, what preview mode does is it subsamples every 25th pixel to allow us to see where our expression is coming from. And this is one area of breast cancer that we're looking at here. So what we did was, when we scanned this in preview mode, we were able to focus on this area where we saw a lot of immune activity specific to myeloid cells in this region. Additionally, in this region, it would be here on this side, but I'm going to highlight it in a separate part, we were seeing a lot of activity of fibroblasts. So cancer-associated fibroblasts were accumulated in this particular area. And so we use cell mode, which gave us that higher resolution to sort of drill down into the biology of this particular area and this area. Now, what makes this interesting, if we think about immunoinfiltration of myeloid cells, or if we're looking at cancer-associated fibroblasts, these are two different regions that a lot of people are studying because of their ability to suppress the immune system. So if I'm trying to develop a therapeutic and I want to understand if my therapeutic is going to be something that I'm going to get an immune cell to respond to cancer, for instance, where we're going to get clearance of that cancer by activating the immune system, these are regions that are doing the opposite. They are suppressing the immune system. And so there's a high degree of interest in regions like this. And so our panel, as designed, is clearly able to detect these regions very easily, and we can do it very quickly using preview mode. You can phenotype these, and you can see in this case, we can sort of do clustering and show you very cleanly that we're able to see subpopulations of myeloid cells and subpopulations of CAFs, as well as our tumor cells, as well as our other immune cells. So we're able to get a lot of information from this panel that you can use to move forward. Also, same analysis, we did a t-SNE. So for those of you who are not familiar with t-SNE, it is a very popular dimension reduction strategy in which we look at three-dimensional space, paring it down to 2 dimensions, and then looking at different subpopulations that are generated based upon input from you, usually surface markers, and we'll look at what's most related and sort of move farther away. So clusters that are closer related as far as expression are going to be a little closer than ones that are less related. In this case, we can see each of the areas on our tissue and exactly where those populations are in our t-SNE. Now, notice there are a lot more colors here than 6, but there are a lot more colors here. So in the t-SNE, we were able to identify all of our markers on this one. So we added our expansion panel kit as well to this. So yes, with 41 markers, this is the level of biology that you're able to quickly achieve. So if you wanted to expand on that with additional antibodies, you of course can do that. Last figure for this particular piece of tissue, the breast cancer tissue, we wanted to show you tissue mode. Tissue mode allows us to scan the entirety of that tissue at 5 micron resolution. And you may be concerned that 5 micron resolution may not be giving you the most information. And so we wanted to sort of address that. Even at 5 micron resolution, we're still clearly able to define T cells, so CD3, CD4, CD8 expression, we're still able to see that. So if we're looking at CD68, for instance, or we're looking at inflammation with iNOS, we're still able to find that. Our hematopoietic cells, all of this is not lost information at this resolution. Now, because it's at 5 microns and not 1 micron, we understand that traditional method of segmentation may not be approachable here. And so what we did as a company is develop a tool for you to help you with this analysis. We used FlowSum algorithms. So these are not algorithms that we designed. We wanted to use algorithms that were vetted and validated throughout science and that people were very accustomed to utilizing. And we developed a tool called an MCD Smart Viewer. And this is something that we give to every one of our customers who has our XTI system that's free of charge to do the analysis of this tissue mode. And what it does is it scans the entirety of that tissue. Well, the first thing it does is you define your markers. You say, in my panel, what are the markers that I want to analyze? And you define the markers, all 41 in this case, or maybe 35, whatever you decide to do, any number or combination, and you simply ask it to cluster those. And it automatically clusters into 8 clusters, 12 clusters, 16 clusters, and 20 clusters. And it generates a heat map for each. And you can utilize those heat maps to sort of figure out which cluster gives you the best information for your particular area of biology. And once you select those clusters, you can then label whatever those clusters are. Let's say there are 2 clusters that are similar and you want to merge them. It gives you the capability of merging that. So you can label everything in your experiment that you stained with the heat map, just like you would pretty much with any other technology. Now, the advantage here is once you've done that, you simply click a button and say analyze. And when you analyze it, it generates a pseudomap of that image. It also generates the capability of generating percentage populations for phenotyping and the ratios in between, as well as nearest neighbor analysis. So it gives you a readout of a lot of information right off the bat in a matter of seconds to minutes. It's very fast. But if you want to take it further, We can change that to a CSV file, and you can put it into pretty much any platform you wish. Now, I took the time to explain that because this is sort of what you're looking at here. We've scanned this entire image, and we focused on 3 different areas of that image that we want to analyze. So A versus B versus C. So if I want to compare one part of my tumor to another part of my tumor, in A, maybe we're looking here at immunodeficient tumor areas, or in B, maybe we're looking at cells that are enriched for T cell activation, et cetera, and so forth. You can read here. Here is our CAF. So we want to focus in C on our cancer-associated fibroblast region. Now I'm going to move into another area. What about T cell exhaustion? So here's an example of how we can high-plex in another area. We can take that same 31 marker panel, but in this case, we're now going to use 5 antibodies from our human T cell exhaustion panel, as well as segmentation, plus 2 antibodies, in this case, CD16 and VISTA. You can mix and match any way you so desire in your panel, but we can add more, and now we're already, again, 41 markers in this panel. In this case, we're looking at an example of human colon adenocarcinoma. Using that same strategy that I talked about before with tissue mode from our XTI system, we were able to analyze this tissue very quickly, and we were able to show you the percentages of each of those areas that we're interested in. Here, the large amount of orange space that you're seeing, I'm sorry, this is more red, I guess. So this red space, this is our immunodeficient tumor area. We're 24% here. This orange space that is pretty much adjacent to it, this is the highly proliferative tumor area of 11%. Now I can contrast that to our tertiary lymphoid structures where we have a lot of immune markers, B cells, hematopoietic markers, dendritic, et cetera, T cell. We see that that's about 1%. Now I'm not going to go through each and every one of these, but you can clearly see how quickly we were able to generate data in each of these areas and provide the percentages. And as I said, this is a matter of minutes. This doesn't take a lot. That software is really just click, wait a couple of minutes at most, and then it's going to give you these percentages based on the areas you ask it to interrogate. Now, we also have the capability of using cell mode, using our segmentation approach. So your traditional strategies of how you've employed analysis, you don't have to rework your workflow. We still have that capability as well. Here's just a mask overlay using our segmentation kit and some additional markers so that we can see that we're getting nice, clear definement of different areas. In this case, we were talking exhaustion, so we can clearly see that we're able to identify CD8 cells that are undergoing exhausted state within this pink region, as well as other markers like TIM3 or PD1. Let's do another example of this. Here, 41 markers. This time we're moving into glioblastoma, the neural expansion panel kit, as well as our neurophenotyping kit, plus our segmentation and our intercalators. Now, this time we're looking at glioblastoma. Now, I've been talking about the speed. This is actually a pretty small section. This is, I believe, the scale bar is 500 nanometers. So this, if we go on the x-axis, this is about 7 millimeters on the x, and I think just a little bit more than 4 millimeters on the y. So if I did preview mode of this entire tissue section, this was about 5 minutes or less to scan, to give you a reference. So very quickly, we're able to scan in preview mode this entire region. And from this, what did we do? We then drilled down on 4 different areas. We wanted to see areas that were rich in myeloids. This is A. We then wanted to look at infiltrating neutrophils. That would be B. We then wanted to look at heterogeneity that was residing within cancer itself. That was C. And then we wanted to look at stemness. So whether or not these clones were differentiating into the same cell type or different cell types, we were able to identify all 4 of these regions with a 5-minute scan, and then we could drill down on each of these regions very, very quickly. So, a scan of tissue mode. This same area of tissue mode, we were able to scan this entire image in about 40 minutes. So just talking about relative speed. And this just highlights how we're able to show different levels of expression from different markers. So we're not losing the ability to see that because we're moving at 5 micron resolution. We're still able to retain that, but we're also showing you a lot of the architecture, so you're not losing any information at all. So if you wanted to be able to define any region, you know exactly that region, and we can then analyze the whole of it or just a part of it, or we can compare one region to another in analysis. And so as I spoke about that, here we can clearly see areas in this dark blue. This is our area of neural proliferative clusters. So about 30% of this glioblastoma. falls into this proliferating state of Ki67 low while expressing SOX2, et cetera, et cetera. Relative to the myeloid, which is this lighter purple, which is about 13.8%. What if we want to look at FOXP3? If you're an immunologist, and then you probably know that FOXP3, in whether you're looking at blood or tissue, usually falls in a ratio somewhere of 1 to 3%. Here we're seeing 2.7% of FOXP3. So our regulatory T cells, in this case, are found in the region on the expected percentage in tissue mode. So we're not losing any information. I really want to make sure I've stressed that to you. Now, I've covered a lot of information, and I hope I haven't gone too fast through it, but I really wanted to stress the fact that we have a lot of tools that are available to you. If you're trying to move into high parameter space, we not only have a system that can do it for you, but we have a lot of reagents and a lot of kits that can move anyone from high knowledge to no knowledge into those spaces.